Good evening to all. Today I am here to discussing about the antigen antibody interactions as a tool for research and diagnosis. So, based on past classes knowledge, we know what is antigen and we know what is antibody. So, the antigens and the antibodies that combine specifically with each other, the interaction in between the antigen and antibody is um, known as antigen antibody interaction. It can be abbreviated just like antigen slash and antibody reactions. If the antigen antibody reactions that occur in vitro and they are known as serological reactions. The antigen and antibody reactions that occur in three stages. In the first stage, the reaction that involves of antigen antibody complex, the second stage that leads to visible events that uh, uh, looks like the precipitation and agglutination. In the third stage, that includes the destruction of the antigen or its neutralization. The properties of antigen and antibody that can be explained with the help of antigen antibody affinity and antibody avidity. So here, what is the antibody affinity and what is antibody avidity and what is called as cross reaction. So when coming to the antibody affinity, the interactions between the antigen and antibody that involve non-covalent binding of an antigenic determinant. In the last class, we also discussed about the epitope. So here the antigenic determinant is nothing but epitope to the variable region. Here the variable region you can see the complementary determining region which is presented on the antibody. So in turn it is also called as CDR of both the heavy and the light immunoglobulin chains is known as antibody affinity. So here the interaction happens in between the antigen antibody that involves the non-covalent binding of an antigenic determinant to the variable region of the both the heavy and light immunoglobulin chains. So what is called antibody avidity? So here the antibody avidity, which is the strength of the bond, the formation of the antigen antibody complexes, it is used to denote that the overall capacity of antibodies to combine with the multivalent antigen. Here the multivalent antigens are of many types depending upon their antigen determinants. If injected into the blood, each antigenic determinant that stimulates the production of the particular antibody. So what is called as cross reactivity? Here it is a immunological reaction that happens with a particular antibody or the T cell receptor that reacts with two or more antigens that possesses a common epitope called cross reaction. So what are the applications of antigen antibody interactions? For determining the blood groups for transfusion of the blood, here we can see the slide agglutination test that is used for the blood grouping is called as heme agglutination, which is also in turn called as the cross match. And the serological assignment of exposure to the infectious agents, tube agglutination is used for the serological diagnosis of the typhoid, brucellosis, and typhus fever. When coming to the passive agglutination test, which is used for the diagnosis of the rheumatoid arthritis, and the development of the immunoassays for the quantification of the various substances like ELISA test, which is used for detecting the AIDS virus. So to detect the presence or absence of protein in the serum, and for determining the characteristics of certain immunodeficiency diseases. And flocculation is the test for antibody detection, depending on the interaction of soluble antigen with the antibody, which results in the formation of the precipitation. And the antigen, which is consisting of the lipid type of particles, for example, VDRL and RBR. So the another one is agglutination inhibition that provides a highly sensitive assay for small quantities of an antigen. For example, in the last class also we discussed about this first home pregnancy test. When coming to the monoclonal antibodies, here what are monoclonal antibodies? Monoclonal, name itself indicating that it is a single type of antibody. The single type of antibody that is directed against a specific antigenic determinant or the epitope. The scientists in the year of 1984, Kohler and Milstein, both are the scientists who achieved for the production of monoclonal antibodies in the large scale. The antibody producing beta lymphocytes with the myeloma cells in vitro that create a hybridoma cell. The polyclonal antibodies are the mixture of heterogeneous, usually produced by the different B cell clones in the body that can be recognized and bind to many different epitopes of a single antigen. The establishment of the hybridomas and the production of monoclonal antibodies, which involves few steps like immunization, cell fusion, selection of hybridomas, culture of hybridoma cell, inoculation of hybridoma cell into suitable host, and extraction and purification of the hybrid monoclonal antibodies. The first one is immunization. In the first step in hybridoma technology, the animal which is used for doing this experiment is the mouse. So that is the first immunized with the appropriate antigen. The antigen along with the adjuvant, which is injected subcutaneously. The adjuvants are non-specific potentiators of specific immune responses. The injections at multiple sites are repeated many times that enable the increased stimulation of the B lymphocytes, which are responding to the antigen. These approaches of the immune stimulated cells for synthesis of antibodies have grown maximum when the serum concentration of the antibodies is optimal and the animal is sacrificed. The spleen is aseptically removed from the animal and disrupted by the mechanical or enzymatic methods to release the cells. A sample of B cells is extracted from the spleen of the experimental animal, whether if we took the mouse, it is mouse, or if we took the Vistar rat, it is the rat, or rabbit, it is the rabbit. So whichever it may be the animal, here we are extracting the spleen of that animal for extracting the B cells. So the fusion of myeloma cells with the B lymphocytes, the extracted B lymphocytes that is added to the culture of myeloma cell from bone marrow. The intended results is the formation of the hybridoma cells that formed by fusion of B cell and myeloma cell. The fusion is done by using the method like polyethylene glycol. When coming to the selection of the hybridoma cells, for selecting the hybridoma cells, the B lymphocytes that containing the ribosyl transferase gene that code for an enzyme like hypoxanthine, guanine, phospho, ribosyl transferase. So the enzyme HGPRT that involved in the synthesis of nucleotides for hypoxanthin, which is presented in the culture medium. Therefore, the beta cells can grow in medium that containing the hypoxanthin, amnopterin, thymine. So here it is called as HAT media, which stands for hypoxanthin, ammonopterin, thymine media. But due to the lack of the HPRT1 gene, the myeloma cells that cannot able to produce the HGPRT enzyme. So, and it does not grow in the HAT medium because of the lacking of HPRT1 gene. The myeloma cell that fused with the another myeloma cell or those do not fuse at all die in the HAT medium. Since they do not utilize the hypoxanthin, similarly B cell that fuse with the another B cell or those do not fuse at all the cells may get eventually die off. So only hybridoma cell, that fused cell between the myeloma and B cell, that can survive in the dividing HAT medium. The screening is done to select the hybridoma cells which are designed a cell for myeloma antibody production. So the culture of a hybridoma cell, the selected hybridoma cells are cultured in the suitable culture medium, often supplemented with the insulin, ethanol, amide, and other additional hormones. The commonly used culture media for hybridoma cell for the production of monoclonal antibodies are Dalbacco modified eagle medium, DMEM medium, and ISCO modified Dalbacco medium, and Ham's F12 4 medium, and Roswell Park medium. Here it is Roswell Park Memorial Institution 1640 medium. So intern is also called as RPMI. After that, the inoculation of the hybridoma cell into the suitable host, these hybridoma cells are then injected into the laboratory animal so that they start to produce monoclonal antibodies. These hybridoma cells may be frozen and stored for further use. Chalo, here we will check the methodology. Finally, the extraction and the purification happens with the help of the method called ion exchange chromatography and antigen affinity chromatography and radioimmunoassay and immune precipitation. These are the methodologies which is used to extract and purify the monoclonal antibodies from the host animal body. So what are the steps here? We observed means here we immunized the animal and took the spleen cells, and we also used the spinner culture after making the myeloma cells, and with the help of the hybrids in the HAT medium, we selected the antibodies, whichever the positive parts here we have, then we freezed that and cloned on the medium. Whichever the positive clones we got, then we characterized such clones and selected the variants and propagated the clones with the help of the medium called different types of medium we have, like a DMEM medium, RPMI medium. So then we injected that clones into the experimental animal where you can see the tumors of the cells producing antibodies. So soon after that, we extracted and purified the monoclonal antibodies with the help of different types of uh, methodologies. So what is the use of this monoclonal antibodies? Means the applications of the monoclonal antibodies. One is the tumor identification. Whichever the labeled monoclonal antibodies we have that specific for various cell proteins are used to determine the tissue source of the tumors by staining histological tumor selection. And diagnostic test, once monoclonal antibodies are produced for a specific substance, they can be then used to test for the presence of that substance in a vessel. This can include like toxins, drugs, or hormones. When coming to the cancer therapy or immunotoxicology, the cancer cells that contain the additional or new antigens on their surface in comparison to the normal cells from which they are derived. If monoclonal antibodies which binds to these cancer cells are isolated, it should be possible to isolate further one or more cancer-specific monoclonal antibodies from them. Such antibodies may form the basis of a new form of cancer therapy, the immunotoxicology. Autoimmune diseases, the monoclonal antibodies that is used for autoimmune diseases, which include infliximab and adalimumab, that effective in rheumatoid arthritis and ankylosing spondylosis by their ability to bind and inhibit the TNF factor alpha. Omalizumab that inhibits the human immunoglobulin IgE, and which is useful in treating the asthma, that is a moderate to severe allergic asthma. And transplantation, there are several diseases where the injection of bone marrow from a healthy individual may be of considerable benefit to a patient. And anti-T cell monoclonal antibody is available and has been used to remove T lymphocytes from the donor a bone marrow prior to its transplantation. The anti-T cell monoclonal antibody, in addition to this, the usefulness in the bone marrow transplantation, which has proved the useful to prevent rejection of the foreign organs such as heart, liver, kidney after transplantation in the body of a patient. When coming to the pure protein purification, the monoclonal antibodies that immobilized by coupling to a cyanogen bromide activated chromatography matrix that is called, for example, like Cepharos, have proved particularly valuable for the purification of the proteins. This is because of the monoclonal antibodies, the antibodies which exhibit the power of discrimination. So finally, we learned about the immunoglobulins, which are soluble proteins that is made up by the B lymphocytes of the plasma cells in response to the exposure of non-self antigens. Each antibody that binds specifically to a single antigen. Antibody molecules have a common structure of a 4-peptide chain. This structure consisting of two identical chains, identical light chain, and two identical heavy chains. The larger chain, CH, on the heavy chain. And so, as we know, antibodies are the glycoproteins. Several different classes of antibodies are differentiated from each of other by their primary amino acid structure. So each antibody class have a specific general function. Depending upon that, there are 5 major classes of immunoglobulins. So here IgG, IgA, IgM, IgE, IgD that differ from each other in size, charge, amino acid sequence, and carbohydrate content. The interaction in between antigens and antibodies, specifically that involves the recognition of an antigenic epitope by a N-terminal variable region called FAB region of antibody molecule if antigen-antibody reactions that occur in vitro, so called as serological reactions. The monoclonal antibodies is a single type of antibody that is directed against a specific antigenic determinant called epitope. George Kohler and Cesar Milstein got the Nobel Prize in 1984 for contributing their achievements for the large scale production of monoclonal antibodies. The monoclonal antibodies are produced by the technology called hybridoma technology. The term hybridoma means is to fuse the cells that resulting due to the fusion of 2 types of cells like a lymphocyte and the tumor cell. So on the other hand, the polyclonal antibodies are the mixture of heterogeneous which are usually produced by different B cell clones in the body. They are recognized and bind to many different epitopes of the single antigen. So our next topic is the regulation of immune function. In the regulation of immune function, yes, definitely we are going to read about the major histocompatibility complex, which is very useful, and the complement system, cytokines, interferons, and humoral immunity and cell-mediated immunity. So major histocompatibility or major histocompatibility complex of genes that consisting of a linked set of genetic loci that encoding many of proteins that involved in antigen presentation to T cells. That most notably the major histocompatibility class 1 and class 2 glycoproteins. Those were the IMHC molecules that are presented as a peptides to the T cell receptors. The significant feature of MHC molecules that presented peptides to the T cell receptor, the special feature of major histocompatibility molecules is their extensive polymorphism. Here the polymorphism is of the critical importance of antigen recognition by the thymus cells. The complement system is a major effector system of the humoral branch of the immune system. It is composed of the plasma proteins in the absence of the infection. These proteins that circulate in an active, which is inactive form in the presence of the pathogens or the antibody bound to the pathogens. The complement system that becomes activated Sorry for the technical glitch. So the complement system that becomes inactivated, the complement protein that interact with each other to form the several different pathways of complement activation, all of which have the final outcome of killing the pathogen. So the complement proteins that interact with each other to form the several the different pathways of complement activation. Finally, the outcome is for killing the pathogen. So the adaptive immunity is of two types, that will be like cell mediated and humoral immunity. The adaptive immunity that generates the antigen specific immune response. Two types of lymphocytes here you can see, like T cell and B cell, that perform cell mediated and humoral mediated immunity. Before going to that, here we have to know about the major histocompatibility complex. Here, the major histocompatibility complex is introduced by the scientists Snell, Dasset, and Benacerraf, the one who received the Nobel Prize in the year of 1980 for their major contributions to discovering and understanding the major histocompatibility system in the mice and human. Here, the major histocompatibility complex is a group of genes that involves on a single chromosome that encodes the major histocompatible antigens. Here, the histocompatibility of antigens are called as transplantation antigens. The major histocompatible antigens are mediated and rejection of grafts between two genetically different individuals. The major histocompatible gene that the gene products are identified as responsible for the graft rejection. So here if we have the major histocompatible gene products that identified and the genes which is responsible for the graft rejection. So the major histocompatible genes, that the products of this major histocompatible that control the immune responses are called as the immune response genes. The immune response genes that influence the response to the infections. If the body is exposed to the prolonged infections, yes, definitely you can see the products of the major histocompatibility genes. So here the major histocompatibility is located on the chromosome number 6 in the humans, that is extends over at least 4 million base pairs. In humans, the one which contains more than 200 genes. In human genes, these genes are called as human leukocyte antigen. Is another name for this one is HLA genes, because these are the genes were first discovered through the antigenic differences between the white blood cells from the different individuals. The essential role of the HLA antigens that lies in the induction and regulation of immune response and the defense against the microorganisms. The monozygotic human twins that have the same histocompatibility molecules in their cells, and they can accept the transplants of tissue from each other. The histocompatibility molecules of one individual that acts as antigens when introduced into a different individuals. The physiological function of major histocompatible molecules that is presented of peptide antigen to T lymphocytes, the major histocompatible antigens and their genes that can be divided into two classes. Here, the first class is MHC1 and the second class is MHC2. The first class, which is a gene that is having the complex which contains the three loci, those were HLA-A, HLA-B, HLA-C, each of which that codes of alpha chain polypeptides. So the second gene complex, like MHC2 gene complex, that contains at least three loci, HLA-DR, HLA-DP, HLA-DQ. So each of these loci that codes for one alpha and a variable number of beta chain polypeptide. So when coming to with this knowledge, our next topic is complement system. So complement is the term which is used to describe a group of serum and cell surface proteins, which is discovered by the scientist Julius Bordet in the year of 1890. Here the complement system are glycoproteins that are synthesized mainly by hepatocytes of the liver, and a significant amount of complements are produced by macrophages, monocytes, and epithelial cells of the gastrointestinal and genitourinary tracts. These components that constitute the 5% of the serum globin and that circulate in the serum. The complement proteins are functionally inactive forms of proenzymes. So here these are the inactive form of proenzymes which are inactive until the proteolytic cleavage happens. Due to the proteolytic cleavage, inhibitory fragment cleave and remove leads to exposes the active site. The complement reaction sequence that starts with an enzyme cascade in the presence of antigen or of antigen antibody reaction, the complement system that becomes activated, the complement protein that interact with each other to form several different pathways of the complement activation. Here when coming to the complement protein nomenclature, that is designated by the numericals like C1, C2, C3, C4, C5, C6, C7, C8, C9. Like that by letter with the symbol and the factor D, or by trivial names like homologous restriction factors. The inactive proenzymes have a simple number designation, for example, like C1 and C2. They were named order of their discovery rather than the sequence of the reaction. So here it is a classical pathway. The sequence is a classical pathway from C1 to the C9. The peptide fragments formed by activation of the component and denoted by small letters. In most cases, the smaller fragment that resulting from cleavage of a component is designated by the suffix A, and the large fragment designated by the suffix B. For example, like a cleavage of C3 produces a small protein fragment called C3A, and the remaining large fragment C3B. By convention, the larger fragment for other factors that is designated by the suffix B, with one exception. For C2, the large fragment was named C2A by its discoveries. The large fragments bind to the target near the site of activation, and the smaller fragment diffuse from the site and can initiate localized inflammatory responses by binding to specific receptors. The complement fragments that interact with one another to form functional complexes. Those complexes that have enzymatic activity are designated by a bar over the number or symbol. For example, C4B2A and C3B, the capital B and B. Here it is the activated complement components that sometime designated by the horizontal line, for example, C2A. So here we have that three types of complement activation. They were like classical, alternative, and lectin pathways. So here the classical component pathway, the classical complement pathway is the pathway which is activated by the immune complexes. Here the immune complexes are nothing but antigen antibody complex. So an antibody antigen complex with IgG or IgM immunoglobulins, which bind to the first component of C1. In addition, the endotoxin, here the endotoxins are the toxic substances which is produced by the gram-negative bacterial cell walls. And certain viruses may directly activate the classical pathway. So the complement component C1 that consisting of the recognition protein C1q that contains that comprises 6 subunits with globular heads and associates with 2 molecules each of C1s and C1r. The classical pathway which is initiated when the recognition of protein that is associated with the proteases either recognizes a microbial surface directly or binds to antibodies already bound to a pathogen. The formation of an antigen antibody complex that induces the conformational changes in the Fc portion of the IgM IgG molecule that expose a binding site for the C1 component of the complement system. Here the C1 in the serum is a macromolecular complex that consisting of C1q and 2 molecules each of C1r, C1s that held together in a complex like C1q or 2s2 that is stabilized by the calcium ions. That binding of C1q2 Fc binding sites that induces of conformational change in the C1r that converts the C1r to an activate uh, serine protease enzyme. The C1r which then cleaves the C1s to a similar active enzyme C1s. The active enzyme C1s that having the two substrates C4 and C2. Here the C4 component is a glycoprotein that containing the 3 polypeptide chains alpha, beta, gamma. The C4 is activated when active enzyme C1s hydrolyzes a smaller subunit C4A from amino terminus of the alpha chain. When exposing to a binding site on the larger fragment C4b, the C4b fragment that attaches to the target surface in the vicinity of the C1 and the C2 pro enzyme then that attaches to the exposed binding site on the C4b, where the C2 is then cleaved by the neighboring active enzyme C1s, the smaller fragment C2b that diffuses away. The resulting C4B2A complex is called as classical pathway C3 convertase. That refers to its role in converting the C3 into an active form. So here the another way is alternative pathway. In this alternative pathway that initiated by spontaneous hydrolysis and activation of the complement component C3. Here the C3 that can bind directly to the microbial surfaces. In this pathway, like alternative complement pathway, that activates the complement and involves four serum proteins like the C3, factor B, factor D, and properdin. And the C3 that exists in two molecular forms, the native form which circulates in the serum that conformationally alter the form in which the thiol-ester bond that have been hydrolyzed. This altered C3 that can bind another factor in the presence of magnesium ions, the factor B of the alternative pathway in the presence of factor D. Here the factor D is a serine protease. So the factor B that may cleave that gives rise to the fragment B, small b together with the C3B form. So the alternative pathway C3 convertase. Here the C3 convertase, uh, the in the classical pathway C3 convertase. Here in the alternative pathway C3 convertase is C3 small b and capital B and small b. So the final complement pathway is lectin pathway. Here the lectins are the proteins that recognize and bind to the specific carbohydrate targets. The lectin pathway is initiated by soluble carbohydrate binding proteins like mannose binding lectin, MBL, and picolin. Here the mannose binding lectin is an acute phase protein that produce in an inflammatory responses that function in the complement pathway, which is similar that of C1q, which is responsible or resembles in structure the mannose binding lectin that binds to the particular carbohydrate structure on microbial surfaces that includes Salmonella, Candida albicans, Listeria, Cryptococcus neoformans, Neisseria strains. After MBL that binds to the surface of a cell or pathogen, the MBL associated serine proteases like MASP1, MASP2 that bind to MBL. The MASP1 means MBL associated serine proteins 1 and MASP2 proteins that have the structural similarity to the CLR and CLS, the one which mimic their activities. The active complex that formed by this association that causes the cleavage and the activation of C4 and C3, the resulting C4B2A complex that called C3 convertase. The C3 convertase is formed. These three pathways that converge at the central and most important step in the complement activation. When any of these pathways that interacts with a pathogen surface, the enzymatic activity of C3 convertase is generated. There are various types of C3 convertase depending upon the complement pathway activated. The multi-subunit protein with a protease activity that cleaves the complement component C3. The C3 convertase is bound covalently to the pathogen surface. Then it cleaves the C3 to generate large amount of the C3b, the main effector molecule of the complement system, and C3a, a small peptide that binds to specific receptors and that helps induce the inflammation. The cleavage of C3 is the critical step in the complement activation that leads to the directly or indirectly to all the effector activities of the complement system. So here the C3b that can also bind to the C3 convertase, which produced by the classical and lectin pathway and form another multi-subunit enzyme called the C5 convertase. This cleavage C5 and liberating the highly inflammatory peptide, the C5a, and generating C5b. Here the C5b that initiates the late events of the terminal pathway, that complement activation in which additional complement proteins interact with the C5b to form a membrane attack complex on the pathogen surface, that creating a small pore in cell membrane that leading to the cell lysis. The first step in the membrane attack complex formation is the C6 binding to the C5b on cell surface. Then C7, C8, C9 that bind to form C5, B6, 7, 8 complex that also known as membrane attack complex. And here you can see the major steps like amplification, opsonization, inflammation, cell migration, activation, and cell lysis via pore formation. So what are the functions of complement system? Here is the lysis of cells, the bacteria and the viruses, opsonization, which promotes a phagocytosis of particular antigens, binding to specific complement receptors on cells of immune system that triggering the specific cell functions like inflammation and secretion of immunoregulatory molecules. The immune clearance, which removes the immune complexes from circulation and deposits them in the spleen and liver. So another small proteins which are having the activities like which acts like a stimulus, like cytokinins are the small proteins that are released by various cells like lymphocytes, monocytes, and macrophages that usually in response to activating the stimulates that induce the responses through binding to specific receptors. Cytokinins act transiently and usually at a short range like an endocrine and autocrine and paracrine manner. Although circulating interleukin 1 and interleukin 6 that can mediate the releasing of complement protein from the liver, the term cytokine that encompasses those cytokines that secreted by lymphocytes substances formerly known as lymphokine and those secreted by monocytes and macrophages, the substances formerly known as monokines. Many cytokines that are referred to as interleukins, the name which is indicating that they are secreted by some leukocytes that acts upon other leukocytes like cytokines. So when coming to the properties of the cytokines, here it is having the high affinity with their receptors. This exhibit induction of the different attributes like a cascade formation, the action of the cytokine on target cell that induce that the cell to produce one or more other cytokines, which in turn induce other cell-to-cell production of the cytokines. When coming to the redundancy, two or more cytokinins that mediate similar function, and pleiotropy, the cytokines has different biological effects on different target cells. Synergy, the combination of two cytokines on cellular activity that is having a larger effect of individual cytokine. Antagonism, the effect of one cytokine that inhibits the effect of another cytokine. The target specific, here the cytokines that binds to specific receptor on a cell membrane of targeted cell that triggers the signal transduction pathway that ultimately alter the gene expression in target cell. When coming to the autocrine activity, the cytokines that can act in an autocrine manner that affecting the behavior of the cell that releases the cytokine, paracrine manner affecting the adjacent cells, endocrine manner affecting the distant cells, although this is depending upon the ability of enter the circulation and on their half-life in the blood. When coming to the chemotaxis, the cytokines that are released by tissues in earliest phases of infection are members of a family of chemoattractants. The cytokines that also known as chemokines, these small proteins that induce the direct chemotaxis in nearby responsive cells that resulting in the movement of the cells towards the source of chemokines. The one which regulates the hematopoiesis and also heals the wound, and this induction of inflammatory responses and the stimulation of the development of cellular and humoral immune response, a control of cellular proliferation and differentiation. These were the biological functions of cytokines. When coming to the classification, here depending upon the receptors, the cytokines are classified into 5 families, like immunoglobulin superfamily. It plays the role in the inflammation. These are induced receptors for interleukin 1, MCF, MCSF, c-KIT, interleukin 18. The other family is interferon family. Here there are multimeric receptors that is composed of heterologous subunits, and receptors are mainly for interferons. This family receptors for interferon alpha, beta, gamma, and interleukins 10, 22, and tissue factors. The chemokines, those were the receptors are G protein coupled receptors with the 7 transmembrane structure and coupled to the G protein for signal transduction. Tumor necrosis for TNF family. These families share a cysteine-rich domain formed of 3 disulfide bonds surrounding a core motif, creating an elongated molecule. The tumor necrosis factor is associated with the procapsis through adaptor protein that can cleave other inactive procapsis that trigger the cascade that reversibly committing the cell-to-cell apoptosis. The final family is hematopoietin family. Here it is a majority of the cytokinin receptors that belongs to this class. These receptors have certain conserved motifs in their extracellular amino acid domain that lack an intrinsic protein tyrosine kinase activity. This family that includes the receptors for interleukins, beta subunit, and interleukin 3, 4, 5, 6, 7, 9, 11, 12, and GMCSF and GCSF, EPO, LIF, CNTF, and also the receptors from the thrombopoietin, prolactin, and the growth hormone. Here the interferons is identified and discovered from the scientist Nagano Kojima, who is from Japan, as a virus inhibiting factor in 1954. It was also discovered by the Isaacs and Linderman in 1957 as a substance responsible for virus interference. The interferons are a family of cytokines, mediators physically involved in altering the cellular immune system through viral infection of the host cells. The interferons not only exhibited important antiviral effects, but also exert a key influence on the quality of the cellular immune response and amplify antigen presentation to specific T cells. The type 1 interferon, like INF alpha, INF beta, is secreted by virus infected cells, while type 2 immune or gamma interferon is mainly secreted by T cell, the natural killer cells, and the macrophages. And interferons interact with the specific cellular receptors, which promote production of second messengers, ultimately leading to the expression of antiviral immune modulatory gene. The functions of there are many functions of the interferons like anti-tumor effect, inhibitory effect on cell growth, stimulation and inhibition of antibody production, and inhibition of delayed type of hypersensitivity by T cell, inhibition of transplantation immune response by T cell, inhibition of blast genesis and DNA synthesis, and potentiation of killer T cells, and potentiation of natural killer cells, and induce of ADCC activity, stimulation of phagocytosis, and the adherence, the potentiality of adherence to tumor cells, the increasing of expression of MHC cells classes 1 and 2 antigens, the inhibition of intracellular bacterial proliferation, the potentiation of NBT reduction in the neutrophils, the increased histamine release in basophils, the promotion of differentiation of erythroblast, induction of differentiation of neuroblastoma cells, the chemotaxis for neutrophils. In the last classes, we discussed about the humoral and cell-mediated immunity. Even now also, we have the humoral immunity is responsible in mediating the antibody molecules that are secreted by the plasma cells, so called as antibody mediated response. The extracellular spaces are protected by the humoral immune response in which antibodies produced. It involves the B cells that recognize the antigens or pathogens that are circulated in the lymph or blood. So, in case of cell-mediated immunity is due to the direct action of the T cells and response to any cell that displays aberrant major histocompatible markers, including cells invaded by all foreign particles, pathogens, tumor cells, or transplanted cells. The T cells have evolved to protect against the intracellular microbes and certain viruses and some bacteria to help the B cell. The response against the extracellular microbes, the self cells like APCs that displace the foreign antigens bind to the T cell. Interleukins, the co-stimulate activation of the T cells. If the MHC1 endogenous antigens are displayed on the plasma membrane. The T cells that proliferate are producing the cytotoxic T cells, the cytotoxic T cells that destroy the cells displaying the antigens. If the major histocompatible 2 and the exogenous antigens are displayed on plasma membrane, the T cells that proliferate, producing the helper T cells that release the interleukin, which stimulate the B cells to produce the antibodies that binds to the antigens to stimulate the non-specific antigens like a natural killer and macrophages to destroy the antigen. So called as cell-mediated immunity. So this is how the cytokines and interferons and humoral immunity and cell-mediated immunity happens in our body. The next class we will discuss about the immune system in health and diseases. Thank you all. I'm signing.